And so for that, we had, this study was born in 2008, just about a year after some very new technologies became available to study DNA methylation. Prior to that, DNA methylation was being studied using candidate gene approaches or very labor-intensive approaches. And so microarray platforms became available that really revolutionized these kinds of studies and made them a lot easier. And there are three types of ways you can study DNA methylation, or three types of basic principles. One involves bisulfite conversion, one involves chopping up the DNA with enzymes that recognize methylated sequences and other enzymes that recognize non-methylated sequencing. Or you can use antibodies to actually immunoprecipitate or pull down methyl cytosines, or use proteins that bind to methylation to pull down the complex. And then you can then use those DNA preps to either apply them to a various number of array technologies and now to next generation sequencing technologies. So at the time, we were using the same sample set that the MMRF Genomics Initiative used, and that was 250. And when you're considering platforms, there are lots of things that go into play. Of course, cost is one of them, but also the type of DNA that you have, meaning the quality, the integrity of the DNA that you have, and the quantity, how much do you have. And so we ended up picking the method that allowed for, because the DNA quality that we had wasn't the greatest, so we needed something that was going to be able to incorporate that kind of DNA, and we didn't have a lot of it. So we ended up picking a platform, I don't know if I have a slide here, but a platform called the Golden Gate Assay for Methylation, and it's developed by Illumina. And essentially that's just an assay that decodes converts an epigenetic code into a genetic code. So you have to remember, one of the complex things about DNA methylation is that if you have a nucleotide of a C and a G, whether it's methylated or not, the sequence is the same, right? So it makes it very difficult. So we trick the DNA. So there's a process of bisulfite conversion that allows you to keep all the methylated C's as C's and change all the unmethylated C's to T's. And so you have now basically a SNP discrimination. You can easily discriminate with that using using the available genomics techniques. So there are numerous methods to study DNA methylation, and this is just to give you a picture of, this isn't even all of them, but this is just a handful of them. This is the number of samples that you can analyze with the assay, and you map that against the number of CpGs analyzed per sample. It shows you that some assays are really, really good at pushing through a lot of samples, but you're dealing with one or two genes at a time. And in the samples the assays where you can study every single CpG change, DNA methylation change in the genome, you can maybe only do just a couple of samples at a time. So there are pros and cons and cost benefits, and what's the type of question that you want. It gives you what will help you decide which assay to take. And this is where my platform fell. And just to give you an idea of how fast the world is moving, 2008 is when we started. This platform is gone. It's passé. It's old news. It's something that is it's a fast-moving field. Okay, so I've kind of talked about that already. And so in this platform, to give you an idea of the resolution, you're able to interrogate per sample over 1,500 CpG spots. So we can detect the changes of 1,500 CpGs in the genome, and those CpGs are found in genes that code for tumor suppressor genes, oncogenes, cell cycle control, apoptosis, a lot of cancer-related genes. Okay, so that's what we have. So what were our results? Well, one of the first things we found was that when we calculate differential methylation, so we compared tumor cells to normal cells, we found that the number of changes increased as you progress through myeloma, okay, which is not a terribly surprising thing. Okay. And this is just a Venn diagram showing from MGUS to smoldering to myeloma that the number of changes increase. There are some unique changes, and then there are some common changes. if you look at the overall methylation values for the genes that were deemed statistically different between normal cells and tumor cells, we found that there was a general decrease in the methylation levels as you progressed through myeloma. So again, myeloma field had focused on hypermethylation, increases in methylation changes compared to normal. And here we're seeing, when you look at a large number of genes, that the trend actually is in the opposite direction. There tends to be a hypomethylation change in the hypomethylation direction. So multiple myeloma progression is associated with hypomethylation. And you can, this is just another diagram to show that, you increase the number of hypomethylation events as you progress through myeloma, but the number of hypermethylation events are actually very frequent from this study. And the really nice thing about this study was that it was corroborated by another group at the same time. In fact, we both presented at ASH, I guess it was 2009, and I guess I was anxious to hear what he had to say, and he essentially presented the exact same story with slight differences, but the take-home message was very much the same. Interestingly enough, that if you look at cell lines, so human myeloma cell lines, so these are cells that are propagated in the lab, the profile looks very, very different. And for a scientist in the lab, this is very important because it brings to question the model systems that we have to study these DNA methylation changes in the lab. So that's what we have to deal with. This may be overkill, but this is just to show that, so if you break up the CPG into islands, or clusters, large clusters and non-clusters of CPGs, most of the hypomethylation that occurred in the non-clusters. So again, in the areas where people weren't really looking in the past. People were mostly looking at changes that happened in these clusters. Okay, these are just a couple of slides to show that this data was advised by whether a patient was treated or newly diagnosed, because we had both types of samples in our cohort. It was independent of hyperdiploid status. We heard Dr. Stewart talk about a patient who is hyperdiploid versus non-hyperdiploid. So they seem to be, in general, these changes were independent of common genomic aberrations that we normally hear or know of in myeloma. Now, these genomic assays allow us to look at how samples relate to each other and how other genes relate to each other within a group. So you can see that, and it might be hard to see, but if these are the normal plasma cells at the far right of the screen for both for M-desmoid and myeloma, you can see that the portfolio for the tumor sample is very much different. So this just gives you a bird's eye view of what's happening. It allows us to possibly identify subclusters of patients that may group together and have unique characteristics, so it doesn't clump everybody into one thing. And so we found some changes in some interesting genes. There was progressive hypomethylation in this gene. This gene is involved in cell adhesion and stabilization of laminin networks and collagen networks in the cells. And this type of pathway actually was seemed to be very important and is regulated by epigenetics. And I won't get into too much detail, but we're able to possibly get a little insight into the biology as well from these types of studies. So what have we learned? Okay, we've learned that hypomethylation is a mechanism of disease progression. That's something that was unknown prior to this study. We learned that there may be, and I didn't really talk about it, but there may be DNA methylation signatures present in myeloma cells that we may be able to interrogate further. Again, this is a point that I hadn't just discussed, but one of the things that we always think about with DNA methylation is how it regulates gene expression. Well, it seemed that there was very little association to DNA methylation changes in gene expression. So it tells us that it's a very complex system that we're working with. And where do we go from here? There are obviously other technologies to think about and more specific questions to ask in terms of interrogating the DNA methylation, but certainly also thinking about some of those other histone epigenetic modifications that may be involved when we're starting to do some of those experiments in the lab. And with that, I'd like to end and thank the colleagues at TGen and the MMRF and the Mayo Clinic. Thank you very much.